The next topic under genetic engineering is that of restriction enzymes. You remember in the introductory lesson I asked you to give it a thought what it would be, what it could be, and if you have studied earlier then you could have revised it very well. Now the name tells that some sort of restriction is being brought about. It would be an enzyme that would bring about digestion of something, that means breakdown of something, and it would be bringing about a restriction. Now whose restriction, who is putting the restriction onto what? You have to remember that prokaryotic cells, or otherwise called bacteria, they have these special class of enzymes which are nothing but nucleases. So first thing is to be remembered that these restriction enzymes are nothing but they are those enzymes which cut nucleic acids, or you can say nucleotides. They are going to cut the nucleotides. So restriction enzymes are nucleases which are found in prokaryotic cells or certain bacteria in order to restrict viruses. Now we get what the term had hidden inside it, that these enzymes are restricting the growth of those viruses which infect the bacteria. So it is a sort of defense mechanism that is found in prokaryotic cells. The bacteria has a defense mechanism which has restriction enzymes which are nothing but the nucleases which cut the viral genome. And once the viral genome has been cut, it is not going to infect. So certainly these restriction enzymes are targeting bacteriophages in specific way. Now this was what was hidden in the name. What is their use when we study it in terms of genetic engineering? These are nucleases. These restriction enzymes are special class of nucleases which cut the genome. There are two types of restriction enzymes. One is endonuclease. Again, I would say these are types of nucleases only. So we have restriction endonucleases and restriction exonucleases. Now from your knowledge about enzymes, you must be knowing that this is going to cut the nucleotide sequence or the polynucleotide or DNA from between, and exonuclease is certainly going to cut it from sides. Exonuclease, outside, in between. So this is what endonuclease and exonuclease difference that we have to study. What is the point of difference between endonuclease and exonucleases? Now when we talk about the genetic engineering, our point of concern is restriction endonuclease, because we have to make sure that in the end we get recombinant DNA where an alien gene would be incorporated into the vector. So we want that the genome is cut in from between. So we are going to deal with endonucleases. Our point of interest is restriction endonuclease. Now what this restriction endonuclease does? It is moving on the DNA. It is inspecting it. Supposedly this is the enzyme. It is inspecting the DNA. It finds a site where it has to cut the DNA. That specific site is known as recognition site of the enzyme. So it first of all inspects the entire length of DNA. It finds the recognition site where it can cut the DNA. The enzyme can act on that particular DNA sequence, and that is known as recognition site. For an endonuclease, restriction endonuclease, the recognition site has to be a palindromic series. Now what is a palindromic series? First of all, what is a palindrome? You know what is a palindrome? The word that is read from front and backwards, it is same. Supposedly we have race car. Okay, race car, read it this way. RACECAR, race car. Read it this way, again it is race car. Now supposedly you have some friend whose name is Nitin. Now Nitin name is also a palindrome. Read it this way, then also you get Nitin. Read it backwards, then also you get Nitin. Okay. Otherwise we have the word Malayalam, the most common example. But I don't remember this spelling very well. Malayalam. I hope it goes well. Malayam. Yes, this is another example of palindrome. Read it this way, then also Malayalam. Read it this way, then also Malayalam. So these are palindromic words. When I say palindromic series, because we are dealing with DNA over here, palindromic series will be such an arrangement of DNA strand in such a way. Now supposedly 5-3 and 3-5, if I have the sequence in 5-3- particular sequence in 5-3- strand, same would be in this if I read it in opposite direction. And the simplest example that we have as a palindromic series on which most of the enzymes go and work is this. Now supposedly this is 5-3-. So you know, according to the law of complementarity, you are going to get this. A would be complementary to T, A to T, T to A and G. Now read it this way, it is G-AA-T-T-C. Read it this way, then also it is G-AA-T-T-C. Now this is a palindromic sequence on the DNA. Of course there would be triple bonds, double bonds, double bonds and triple bonds, something like this it would be. Now what happens is this restriction endonuclease identifies this palindromic series as the recognition site. If that nuclease is for this series is the recognition site for the given endonuclease, then it comes over here, it cuts it like this I would show. This is the backbone being cut. I would use red. It cuts the backbone between T and C, then it breaks these bonds and cuts it like this. Now these bonds are broken. What do you get is this. Okay, same way you get like this. You have got sticky ends. These protruding sequences are known as sticky ends. Now you are going to get the DNA which has been broken like this and it forms the sticky end. Suppose this was the sequence, so you get one series like this which is not having anything over here, and this one becomes like this, it is not having anything over here. So you get these sticky ends. Now supposedly you have to fix something into these fingers. Supposedly you have to fix something into these fingers, so you need to have some place to fix, then it will come and fix over here. So these places, these crevices which are created, in the same way it creates a sticky end so that the gene of interest can be switched into here and it could be fixed over here. So this is what is being created by restriction enzyme. Firstly, it sees the recognition site which has the palindromic series. It cuts that palindromic series in such a way that sticky ends are being created. This is the purpose that restriction endonuclease serves. Exonucleases, they are going to attack the endpoints and they are not going to create the sticky ends. Now supposedly this was to be cut by an exonuclease. It would have simply cut it this way. You are not going to find sticky ends, instead you find blunt ends. Okay. So this is all you have to keep in mind when we talk about restriction enzymes. We do have another lesson where we see how restriction enzymes are named. What are the different types of restriction enzymes, a short lesson. You just have to remember those things which would be discussed. But for now, you have to understand what this enzyme is, how it works on a particular genetic sequence, or rather I would say a DNA sequence, how the DNA is being cut and sticky ends are being obtained, how the counterpart of these endonucleases produces the blunt ends, and what is palindromic series. I would suggest you to remember as many palindromic series as you can for specific endonucleases, but for that you need to watch the next lesson first, where you get an idea that such is the naming process of endonuclease and what could be its palindromic series.